ওয়ার্ল্ড মাইক্রোবায়োম একটা পিপিটি আছে।
The eve of World Microbiome Day. So, we are gathered here today in the seminar hall to celebrate a world within worlds, the invisible yet powerful universe of microorganisms that shape our health, our ecosystems, and our understanding of life itself. Each year, this day reminds us of our interdependence with the microbial world. And today's event promises to take us on an insightful journey through that invisible realm. To begin our program, I would like to invite Dr. Shovik Mukherjee to deliver the welcome address. Sir, to you.
So welcome all to the World Microbiome Day Symposium. And as you know, from 2022, this World Microbiome Day celebration has started in different parts of the world. And this is the fourth mini symposium. The third mini symposium last year was amalgamated with our national workshop on microbiome. And this is the fourth mini symposium where we have invited our speaker. So before going to the lecture of our speaker, let me just give a brief introduction, because every year the audience is not same. Students do join and they enroll themselves. So what is the implication of the World Microbiome Day? So World Microbiome Day, as you see, we are celebrating the invisible world of microbiomes. And why we call them invisible? As you know, these are microorganisms. Actually, the World Microbiome Day is on 27th of June, but for obvious reasons in this year, we are celebrating it on 26th. Uh, Microbes are our constant companions. As you can see, we are in NIBMG, we are more focused on working on human microbiome, but microbiome is everywhere. And as you can see, it is present in soil, in plant, in different parts of the environment. You can see that in the fish, in the aquatic environment, agriculture, everywhere the microbiome is present, and it is very important. Microbes are also essential for living beings, and it helps the human health. We are all, some of us are working on human microbiome. The immunity, the mood swings, and brain-gut axis, skin-gut axis, you name it, and we have it. And microbes also play a critical role in both human and environmental well-being. And not only for humans, for the sustenance of life on this planet, we need the balance of the microbes. So without further ado, we will just start celebrating the mini symposium on this World Microbiome Day, and let us all join the microbiome movement. Now, today's talks are, the first talk will be of our speaker, Dr. Shulagna Basu. She is the scientist G of the ICMR National Institute for Research on Bacterial Infections. This is, the name of the institute is very new, but actually we are all familiar with the previous name of the institute. It's called National Institute of Cholera and Enteric Diseases. And she has done her PhD from PGI Chandigarh, and then she has joined in this institute, and her major work area is working on AMRs in the neonates, and she is more interested in seeing the transmission of the AMR microbes from mother to neonates. And this is what we were talking just before this, coming to this seminar hall. We were interacting, and she was just telling about how she identifies the AMR bacteria from the stool samples, and then look into their transmission and do the isolated genomics. Rather than doing the microbiome, she does the isolated genomics of these bacteria and identifies very interesting things. So without, after that, I will speak on the another program that our institute is a nodal center for, that is the One Day, One Genome program. And then Shankho will give a flash talk on one interesting research that pertains to the area that we are in. In Nadia area, the arsenic contamination is huge, and how does it affect the gut microbiome? So without further ado, I will just invite Dr. Sulagna Basu to deliver her Microbiome Day lecture in NIBMG. Over to you, Dr. Basu.
কিন্তু দাগাবো কোথায়? This is the only problem with this one. এটা লাগিয়ে রাখো. Okay.
In case you need.
I will need it now. Can you just open up the presentation for me? This is the one. Good afternoon, and a very good afternoon to all students, scientists at NIBMG, and thank you so much for inviting me here, Dr. Shobhik. It's a pleasure to be here. And so before I start my talk, my laboratory primarily focuses on antimicrobial resistance. So there are several aspects of resistance that we study. And in today's talk, I will be focusing on the transmission of resistant genes from the mother to the neonates. And that is the reason why it is titled Tiny Lives, Big Threats, Transmission of AMR Genes. So there are several aspects of this talk, and before I go into my work, I will have a brief introduction of the problem of neonatal infections, the problem of AMR, and the specific genes that we will discuss today. So before we go into the area of problem, so what is the good that is happening around the world now? So this is from Hans Rosling's assessment, where he shows that there are 16 bad things which are decreasing. So oil spills have lessened, child labor is going down, and in terms of health, smallpox is no more. And you can also look into this part. There are much lesser children dying these days than they were earlier. But in spite of all the good that is happening around the world, there is an underlying problem, something which we all know, but we only recognize when we land up in a hospital or when our loved ones are in the hospital. And that problem is the problem of antimicrobial resistance. So antimicrobial resistance, if you see, is like a ticking time bomb. So it has been discussed in the World Economic Forum and also at several high-level meetings at WHO and also the UN General Assembly, that arguably the greatest risk to human health comes in the form of antibiotic-resistant bacteria. We live in a bacterial world where we will never be able to stay ahead of the mutation curve. A test of our resilience is how far behind the curve we allow ourselves to fall. So just as we are celebrating the Microbiome Day, we also I have taken this opportunity to tell you about how much or how prevalent AMR genes are in the human gut. And that too in the regions that we live in or in India where this problem is much more than in other parts of the world. Now, what is the major crisis? So, it has been predicted in an excellent paper by O'Neill, where he has shown that if we carry on the way we are at the moment, 10 million people will die because of AMR infections by 2050, which is much more than what people will die due to even cancer. So this is a problem, as I said, a silent pandemic. So we are not aware, but it is huge. It's a big, big problem across the globe. Now, how big is the problem? Different statistics, particularly the global burden series of papers, have shown, and you can see here a map of the world. Most of the countries are in orange or in red. All of these actually indicating resistant bacteria at a level of 50 to 60% or more. So several places across the globe are having very, very high resistance. Now, what about the problem at the national level? If you look into this problem, you will see that India has a very high drug resistance index, something around 71, and this was a data from a few years back, so there is a possibility that it is even higher now, and which is the highest across the globe. So we have a huge, huge burden of antibiotic-resistant bacteria. So now, what are the pathogens that we work and we focus on? My laboratory is primarily focusing on, this is a WHO priority list which was updated in 2024. And this list has, as the critical group of bacteria, you have Acinetobacter baumannii, which is carbapenem resistant. Then again, third generation cephalosporin resistant bacteria, enterobacterials, primarily Klebsiella, E. coli, all of these, and carbapenem resistant enterobacterials. And we in our laboratory primarily focus on Acineto, E. coli, and Klebsiella. And we look into resistance mechanisms, again focusing on, as I said, carbapenem, which a part of my talk will be on this particular gene called NDM, and then a part of my talk will be on colistin resistance, which we will discuss the MCR1 gene. But we also do other work on other resistance mechanisms. Again, as I said, so this talk at the central point remains the neonate or the tiny lives that I referred to in my title. So neonatal mortality, if you look into the numbers, you will find that most of the child under 5 deaths happen during the neonatal period, which is a period from birth, that is 0 days to 28 days of life. And most of the deaths are happening in this extremely vulnerable group of patients. Again, why are we concerned about neonatal mortality? India has shown exemplary reduction in neonatal mortality rates, but you can also see that across the globe, the decline of the infant mortality rate has been much more than the neonatal mortality rate. So the decline of the neonatal mortality is much slower, primarily because this is a period where the population gets very vulnerable to infections and mortality is very high. And again, so as I say, not all children are born equal, and most of these child deaths are in countries, specifically India has a huge burden of, as I mentioned, AMR, but it also has a huge burden of neonatal deaths. Also, several other Southeast Asian countries have, and Sub-Saharan Africa also has a very high rate of neonatal mortality. The scenario in India is also, as I said, neonatal mortality rate has reduced a lot, but there is a huge disparity across the several states of India. So if you have a state like Kerala, neonatal mortality is comparable to any developed country. But there are states like UP, Madhya Pradesh, where the neonatal mortality rate is much higher. So there is disparity across the nation. Now we are coming closer to the work that we are doing. And as I mentioned, I will be talking about the vertical transmission of bacteria. So just after birth, the gut of the neonate starts to get colonized. And while the neonate is moving through the birth canal, or even after that, it gets bacteria from the mother. Now the point is, if the mother is already harboring resistant bacteria, resistant genes, then how does that affect the neonate, and what are the chances of the baby becoming sick? So several studies have shown that the gut of the bacteria, particularly in the premature low birth weight neonates who have been in the hospital for a long period of time, there are chances that the bacteria can translocate from the gut through the mesenteric lymph node and cause sepsis. So any bacteria that is resistant moves from the mother to the neonate can pose a lot of problem for the neonate. Okay, so here we are talking of bacteria moving from the mother to the neonate, but we are also in a way discussing the exchange of plasmids or resistant genes on these plasmids within the bacteria in the gut. So we cannot forget the contribution of these scientists. Young Lederberg was working with Tatum, and they were the first, they received the Nobel Prize, and they were the first to show that gene recombination was happening in E. coli, and they also went on to show that exchange of genes can happen between one bacteria to the other. The other person is Stuart Levy, who in 1976 showed that the spread, the first time he could show that antibiotic resistant plasmids were moving from chicken to chicken and from chicken to man. So this was the first study which showed that resistance genes passing via plasmids can become a very worrisome thing. And we know the fact now, it is actually a huge, huge problem. Okay, two particular genes I will be discussing in my talk. One, the BLA-NDM, which is responsible for carbapenem resistance, and the other, MCR, which is responsible for colistin resistance. And both these antibiotics are last resort drugs. So these are only given or should only be given to patients who are extremely serious or who are seriously ill. So these resistance to these two antibiotics always remains a point of curiosity or something that we need to know. So what is the burden of such genes in the environment, in the human gut, or elsewhere? So blood, if you are studying blood infections, that we do, primarily we do that. So we are looking into neonatal sepsis. So we always have a look, are these bacteria resistant to carbapenems? What is the gene that is there? Or also there's a huge interest in MCR. And MCR interest primarily was driven from the environment, because that was a gene which was actually identified first in animals, and then it had moved on to the human population. And that was also because the use of colistin in the poultry farms, in the animal fodders, was very, very high. And that led to the transmission of this gene from the animals to the environment and then into the human population. So when I was talking of sharing of genes, this is a picture which is a little old now, but it gives us a glimpse of how these genes have now spread all across the globe. So you have this NDM gene, which is endemic in India. So you have a lot of these in India, in Pakistan, whereas another resistant gene called KPC is endemic in other parts of the world. So these genes have been identified, maybe NDM was identified in 2010, and within a span of 15 years, it has just spread all across the globe. The other gene which I was talking about is MCR, and it is the same. Again, if you see, it is present in animals, in humans, also in the environment, but it is present all across the globe. Now, this was a study which we were doing, which was titled "The Burden of Antibiotic Resistance in Neonates from Developing Societies." And this was a multicentric study being carried out in different parts of the globe. And one of our centers were in Kolkata. And through this study, we were primarily looking into antibiotic resistance in neonates with a focus on the beta-lactam resistance. So either resistant to cephalosporin or resistance to carbapenems. And this particular study came up and gave us a few important insights, which initially we thought Klebsiella is the predominant organism that causes sepsis in this part of the globe. But this study could show that Klebsiella is also predominant in other parts of the world, particularly causing neonatal infections. And it also showed how resistant genes were present in the mothers and the neonates. But this study was primarily looking into the burden. We took up this study, or we followed this up with studies of how this was getting transmitted from the mother to the neonate. So most of the study that I will be talking about was done in the neonatal intensive care unit and of the IPGMER and SSKM hospital. And this was the Barnard's team. They were actually visiting us at that time, and this is the neonatal unit at the IPGMER hospital. So so the first part of my talk today will focus on colistin resistance or the MCR gene. So colistin is a very old drug. It's not a new drug like the carbapenem. And the use of colistin was actually, it was withdrawn because of its neuro or nephrotoxicity. But now that antibiotic resistance has become so rampant, lately the use of colistin has also increased because there was no other resort and the clinicians had to use this particular antibiotic in spite of its toxicity in humans. Because it was actually withdrawn from human use during that period, as I said, colistin was being used in the animal sector in a significant way. So this had led to the proliferation of this MCR gene. So there are several mechanisms of resistance of colistin, but today, because we are talking of transmission, we will be concentrating on this, on the acquired colistin resistance gene, or which is called the mobilized colistin resistance gene, and we will be focusing on this part. So you can now see that there are several, this happens very, very commonly in bacteria. They evolve. So they start off with one particular variant of an enzyme, and then within a few years you will have more and more different variants that are present. So even MCR, you can see, has several variants now. Now, how does colistin work? Colistin is a positively charged antibiotic. So it primarily interacts with the negatively charged lipid layer, and then it destabilizes the layer and causes bacterial death. Now, this particular gene, MCR, it encodes an enzyme called phosphoethanolamine transferase, and it transfers the phosphoethanolamine at the LPS, due to which this negative charge decreases. And because this is a positively charged antibiotic, the increase of positive charge actually causes repulsion, due to which colistin does not function. So this, as I said, we were carrying out this study, and this study had primarily enrolled pregnant mothers, around more than 1,000 pregnant mothers. And then if the baby was sick, so if the mother had a sick neonate, then the neonate was also enrolled. The gut, the rectal swab of the mother and the neonate was actually taken, and the blood sample of the baby was also taken. And then these were cultured on vancomycin supplemented plates with colistin, because we were primarily interested to identify bacteria that were resistant to colistin. So what was the initial finding? The first thing that we found was that the isolates which were colistin resistant and had this particular gene were found only in the maternal gut specimens, and there were very few of them. So even if MCR has a huge spread across the environment and the animals, its presence in this population was much low, was not very high, but it was present. So these were all healthy mothers who had no complications during birth, but they still harbored this gene. Now, we had collected gut specimens from the sick neonates, but none of these sick neonates were found to harbor the MCR gene. The MCR 1.1 variant was identified, and very interestingly, when we isolated the bacteria, there were other bacteria also present, such as Klebsiella and Acinetobacter or Pseudomonas, but MCR was only found in E. coli. And it was found in all mothers, none of them were related in any way. So this was a gene which was primarily found in Escherichia coli. And in contrast to several bacteria, though these isolates had the MCR gene, the resistance determinants in these bacteria were not very high. So you either had a TEM or a QNR, which indicated quinolone resistance. Several of them had QNRS1, as you can see, but apart from that, their resistance to other antibiotics was comparatively low. The other thing was that we wanted to see, is it a particular clone of the MCR containing bacteria which was spreading, or was it distinct from each other? When we carried out the sequence types, then we found that most of them were distinct. There were a few which matched, but most of them were distinct. And these bacteria also had a number of plasmids. Several plasmids were present, and most of them were very, very large mega plasmids that were present. So this is a pulse field gel electrophoresis which was carried out in the maternal samples. As I said, the neonatal samples did not possess this gene. So we just looked into them, and we found that whatever the whole genome data was indicating, the pulse field gel electrophoresis also indicated that these strains were diverse. And two novel STs were also found, which is found in the gut, and the phylogroups were primarily A and B1, which again indicated that the E. coli which are primarily found in the gut are generally belonging to the phylogroups A and B1. Now we looked into the transmissibility of this gene, and it was found through conjugation that conjugate transfer of this gene was successful. And it also indicated on which plasmid this particular gene was present, and it was present in the ink HI2 plasmid except for one. Now, since, and these were, as I said, were mega plasmids, so they were of the size 216 kb and the other was 240 kb, both having the same replicon type, but they differed in their sizes. And this again was an indication of the promiscuity of this gene. So it could easily shift from one bacteria to the other. Now, since there was no particular clonal lineage that was spreading, we wanted to see whether the plasmids were similar or not. And we had already seen that they were all ink HI2 plasmids. We mapped these plasmids after the sequencing, and we found two different clusters of the plasmids. One, as I said, though they were all HI2, the one group belonged to 216 kb, the other was the 240 kb plasmid. And we also looked into the immediate genetic environment, and we found that most of these plasmids, the genetic environment around this gene was very, very similar. similar. So this, in a way, this particular picture brings about an understanding of what was happening during this study. So there were two modes. You can see that transmission of this gene can take place through clonal, either a particular clone of bacteria that is spreading across the unit or has spread across the society in such a way that these pregnant mothers who were in the unit had this particular clone present. But as I said, most of these strains were of different sequence types, indicating that this was a different lineage. So the mothers probably either acquired the plasmid, a common plasmid, and you can see here that these three mothers had the same ST type and were all in the hospital around the same time. So this was probably a transmission that happened within the hospital. Whereas when we looked into the other different lineages and we looked into the plasmids, we found that a few of them had the 240 kb plasmid, whereas most of the others had the 216 kb plasmid. So there was a chance of this having spread between the mothers. And in some of the mothers, you would also see that there were more than one isolate of E. coli, and these were of different sequence types, but they had the same resistant plasmid. So you can see here this particular case, the 727. So this was one mother which had a 727 R1. Oh no, let's go to the 750, where you have two isolates, both E. coli, but they were different E. coli because they belong to different sequence types, but they had a common plasmid, which indicated that the transmission of this gene was happening within the gut of the mother. Now, a few things that we could see was that there were significant similarities in the plasmic background, but when the plasmid was even similar, there is a possibility. So the way we had carried out the study, we could not conclusively say whether this exchange had happened within the hospital or whether there was an independent acquisition of these plasmids in the mothers at other points of time. Now, we also did a comparison of our strains with the global, the MCR positive E. coli, primarily from Southeast Asia, because most of these MCR positive strains of E. coli have been reported from China, Taiwan, and the surrounding regions. And when we compared these genomes, we did this core genome phylogeny, which could show that the study strains were close to strains from various countries such as Bangladesh, China, Thailand, Singapore, Vietnam. And study strains also were similar to E. coli, not only from human specimens, but from animals, flies, and which actually emphasized the presence of this gene in the environment or within the food chain. Because there's a lot of prevalence of this particular gene in the animal, the poultry and all of that. So that is a possibility that we could see. So this paper was published in Emerging Microbes and Infection, and the few things that I would like to share with you is, in a nutshell, is that this particular gene is not very highly present. It's the percentage of prevalence is low. The variant that we isolated from all different mothers from different parts of the state all showed this only the MCR 1.1 variant. The species was only E. coli and they were present in some novel STs but in other STs too. The carriage of these genes was only seen in the mothers, not in the neonates. And in this study, the limitation was that we did not look into the healthy neonates. The study was designed in a way that we only looked into the septicemic neonates. So that remains a limitation, and we were doing another study with other hospitals where we also included the healthy neonates, and we haven't come up with all our data, but we will be able to understand what is happening there. Also, the plasmid that was bearing this gene was a conjugative plasmid, and these plasmids were similar to plasmids reported from Egypt and Saudi Arabia. The genomes, as I have already mentioned, were from similar to various sources from human, animal, environment. The other important thing was that all of the mothers who were harboring these genes were actually healthy mothers, and they had not received any antibiotic before they gave birth to the child. But the presence of such genes in healthy mothers is actually very worrisome, because these may just go undetected and lead to the spread of resistance. Now, this is the other part of the study on NDM, and if you again see, this is a part of the Barnard's data where these particular genes, the NDM, was present in the neonate, and the rates of resistance was very high. So 42% of the enterobacterials were actually possessing NDM, whereas in mothers, comparatively, this gene was much less. Whereas if you take another gene, you will again find that CTXM, which is resistance to cephalosporin, again shows high prevalence in the mothers, but is much low in the neonates. So we, from this particular study, we took this forward, and we wanted to again see the transmission of these gene or the, not particular the gene here, we were primarily focusing on the transmission of the resistant bacteria from the mother to the neonate. So this is a gene which is created a lot of furor because of its name. It is called the New Delhi metallobeta-lactamase because it was initially identified from a patient who had come here for treatment in New Delhi, and the patient went back and this gene was identified, and at that point of time there was a huge hue and cry about this nomenclature of the gene. But this particular gene, as I said, is endemic in India. So you find now a lot of bacteria and different species, not a particular species like E. coli, but you find it in E. coli, Klebsiella, Acinetto. You also find it in Vibrio, which is in the environment. You find it in most of the species. And this is an extremely potent gene because it can hydrolyze all the beta-lactam antibiotics. So the whole range of beta-lactam antibiotics are hydrolyzed and become ineffective. And that's a huge setback for the clinicians because beta-lactams are used widely for several diseases. Again, a very similar study where we looked into the presence of the NDM, and as I said, it was much more in the mothers, babies, than it was found in the mothers. And primarily that was probably because they were hospitalized, they were septicemic neonates, they were hospitalized for a much longer time in the hospital. Now, from this study, there were 86 mother-baby pairs. So we took only the pairs. In the earlier study, we had screened all the mothers, but here, because the prevalence of the gene was much high, we only took the mothers who had a baby who had sepsis. So we used the mother-baby pairs. And in this case, we had 86 mothers and 93 neonates, because some of them were twins, and we could isolate around 20 NDM positive isolates, and from the babies, there were 42 NDM positive isolates. But when we looked into the species, and we were looking into we wanted to see the transmission of the gene, so we looked into whether there was a common species between the mother and the neonate. And we identified 8 pairs where there was a common organism. So either the mother had an E. coli and the baby also had an E. coli which was NDM positive, or you had a Klebsiella both in the mother and the neonate, and you also had an NDM. So these were the pairs that we studied. And we looked into the diversity of these bacteria again through PFG and whole genome sequences, and we deciphered the resistant genes. And you can see here, in contrast to the isolates that were harboring the MCR, these isolates which were carrying the BLA NDM also carried several other resistance genes. And this is very, very typical of NDM. So when it moves, it is also carrying other resistant genes. So it not only hydrolyzes all the beta-lactam antibiotics, but it also creates resistance to other antibiotics too. And these isolates also had a number of replicons or plasmids. Again, this is a picture of the Klebsiella isolates. It is nearly the same case. You will find that the isolates are very different, but there are several resistant genes along with the NDM that are present. And in case of MCR, we always had an MCR1, but in case of NDM, we either had an NDM1, 5, or a 7. Now we compared these pairs and we looked into, okay. So the other important thing that we noted was that these were isolates in the mothers or the neonates, and many of them were epidemic clones which had these clones here. So these are epidemic clones which are present in the gut of the baby or the mother. And we looked into these pairs and we found that of these different pairs, there was one single mother-baby pair where the isolate was identical. So the mother had a bacteria which was exactly similar to that of the baby. In some other cases also, you had very close sequence types, but they were not the same clone. So this was the only case that we noted. We also noted, so there was this clonal transmission from a mother to the baby, which we, as I showed in the last diagram, and you can see that the environment of the gene around the NDM is also exactly same. The environment, there was another pair, but this was, there was not another pair, this was an unpaired isolate. So there was one mother and another baby isolate which were also clonal. So we had the same clone in one mother and a different baby. But here again you can see that the background of the gene is very, very similar. So again, coming to this part, you can see that the overall carriage of this particular gene NDM is much higher than the MCR. The mother-baby pairs were primarily distinct and distributed within diverse sequence types, but they also included several epidemic clones, which actually is very worrisome, because these clones are are actually a major cause of the spread of NDM resistance to carbapenems and the presence of this gene. Particularly ST11 is very common in China. ST147 is also common across the Southeast Asia. The other thing we noted was that you only had MCR1, but here you had different variants of the NDM, 1, 5, and 7. All of them were on conjugative plasmids. I did not show the result of the conjugation here, but all of them were on conjugative plasmids, either the F2 or the F2K. So there is a spread across these species of these replicons, and that has led to the huge, and this is again a very, the conjugation, the efficiency of conjugation is extremely high in this particular gene compared to, again, the MCR. Now, what about the mother-baby transmission? Now, genomic analysis here supported one case of vertical transmission, but again, we only had taken the mother and the neonate if the neonate was sick. So we only looked into the sick pairs, the mother and the neonate there, and we need to see what is the case of such transmission between healthy neonates and their mother. And in a subsequent study, we are trying to follow up the neonate up to a 6-month period so that we can find out whether the carriage is persisting in the neonate for a long time. The other is that screening of the gut is extremely important, and infection prevention policies are also need to be very stringent, because this particular gene was nearly present in 42% of the neonates, which is very, very high. I am at the end of my talk, and I would like to end with this Walt Kelly's Pogo strip, where Pogo is sitting here and he says, "We have met the enemy and he is us." In case of antimicrobial resistance also, the enemy is us. It is our misuse, non-judicious use of antibiotics in the animals, in the humans. Our infection control practices are not stringent. There's a huge efflux of antibiotics from pharmaceutical companies into the wastewater. Treatment of hospital waste is also not proper, and all of it has led to something that, as I said at the very beginning, it is like a ticking time bomb. So my student Shormi and Priyanka, most of the work has been done by them, and this is a picture of the Barnard's group when they were here visiting us. And apart from them, I would like to thank many of my students and the funding agencies. The Barnard's work was funded by the Gates Foundation, but the subsequent work was funded by ICMR. So thank you all, and I would be happy to take any questions that you feel.
Anyone, if you have any questions, you can ask now, or else we'll move ahead.
Thank you, Dr. Basu. It's a very, very illuminating talk. And this is really truly the enemy is us, and maybe some parts of us. So looking at the data that you showed and the kind of correlation, so of late, have there been any correlations with surveillance measures on AMR, particularly those
Surveillance?
measures where you are looking at wastewater based surveillance of AMR? Have there been any tracking of these genes or these
Several
isolates?
people are tracking these genes. We do not do it because we primarily focus on human health. But I can tell you that I think TIGS is doing some work on wastewater and they do find this. And initially in 2009, when this particular NDM gene was identified, we were the first, it was identified in that Swedish patient first. But after that, it was our study which showed the presence of this gene in a very remote area in Bengal. It was in the district hospital in Shiuri. We were doing a study there, nothing related to this. This was an IPGMER study, but it was found there at that point of time. And after that study, because that was a neonatal thing, there was this other group who had reported this. They found this in the drinking water samples in New Delhi also. And that is the time when there was a huge uproar about the naming of the gene and a lot of mudslinging at the fact that somebody is naming it on a gene with the capital of India and all of that. But since then, a lot of people are doing work in, as you said, wastewater. In hospital waste, you get lots of them. But there are studies now, there's this Ganga project or something, that river project that, huh? Yeah, they are also identifying this gene from certain parts of the.
So in hospital waste, this monitoring has been done. Have these been isolated in hospital waste?
In some studies, I have seen the reports of the BLA-NDM gene in hospital waste. So people have done, but it has not been done that methodically. There are some studies being carried out at one place and then there's not follow up or it's not done in several areas too. But TIGS is doing some work. I am not sure whether they've published their work, but they are doing an extensive study of wastewater surveillance of these genes all across the southern part of India. So several states are.
So since AMR is now all pervading, and as you said that the enemy is us, what is your idea about, or your comment about the bacteriophage therapy that is now emerging as a biotherapeutics on to combat these AMR?
If you tell me, in a way, I am in a dilemma. I know that a lot of work is being carried out in this area, and several case studies have also reported the success of bacteriophage therapy on specific patients. But there is also a lot of, I would say, a lot of doubt and doubt about its of course, there is this therapeutic angle, but there's also a doubt about whether this therapy will become something boomerang or not. So again, there is, but I feel if anybody asks me, I will say that infection prevention and control, your hand hygiene, this hospital that we work with is one of the best, and their intensive care unit is comparable to any developed. They have a very low rate of resistance. So though we isolate organisms because they have a lot of referral cases from several parts, but they have the rates of resistance are low.
Thank you, Dr. Sulagna Basu. And, she is also a member of the WHO Technical Advisory Board on AMR, and a fellow of the Indian National Academy of Science. So, Lakshmi, please come. We will give you a memento as a remembrance of your talk in NIBMG.
This also gives me an opportunity to actually spread the word that we have a repository at NIRBI. Dr. he knows it very well. He's also a part of our advisory group. So if you need to work on any strains, you can write to us, write to me, and then we can share strains with you. And these strains are collected from all across India, not only this part of, so it's a part of the AMR network that ICMR has. So we will be happy to share strains or share ideas or help you in any way if you want to work, because NIRBI now has a huge focus on AMR. And you are also doing a lot of work in AMR. One of your microbiome student is working. There should be more people working so that we see something at the end of the tunnel. Thank you.
আমাদের ইয়েটা কোথায়? স্লাইড স্লাইডটা অন করে না, স্লাইডটা অন করে দাও। আমার 5 মিনিট লাগবে, তারপর তোমার কোথায় এখন না না সব খুলে রেখেছিলাম। কোথায় গেল এইটা? আচ্ছা এটা না হয়েছে, এটা ওডিওজি। Yeah, this will be required. So I'll take only 5 minutes, and we'll try to impress upon the fact that in NIBMG, we are actually the nodal center of something that DBT and this Biotechnology Research and Innovation Council is a flagship project of that. It's called the One Day One Genome. The One Day One Genome project is primarily focusing on the bacterial genomics now, but soon it will extend to fungus and other kinds of organisms. So as we all know in NIBMG about human evolution, right? Out of Africa, coming from different parts of geography and climate, how they shape the human genome. But let me tell you this, that along with the human genome, the microbiome is also co-evolving. And therefore, you see this human exodus and the change from the chimpanzee to humans. And you also find our microbiome has co-evolved and become more and more industrialized now. So the microbes are also on the move. And if we come to India, since this One Day One Genome project is mostly focused on Indian microbial biodiversity, you can see our India as a country has a lot of such biodiversity areas. Like we have these biogeochemical cycles, methanogenesis, soil bioremediation. So the main idea of this One Day One Genome initiative is to fully harness the diversity of the bacteria present in different parts of India. And DBT has started this One Day One Genome initiative aiming to decode and publicly release one annotated microbial genome every day. Now, this is also in sync with the BioE3 policy. You have heard about a mini symposium on BioE3 sometimes back, where we have experts from different institutes. And in BioE3 also this One Day One Genome initiative fits in, because the BioE3 is economy, environment, and employment. And here, this drives the economy driving the biotech innovation. This can also be apart from the focused on microbes, because microbes also produce certain enzymes that can be a more industrialized and more important for the industry. And in environment, it also enables the discovery of the microbes for sustainable agricultural and other pollution control. And by doing this, it always is going to have some employment of individuals in these bioincubation hubs. Now, what is the objective of this One Day One Genome project? The One Day One Genome project's objective is to publicly release an annotated microbial genomic data every day, where the unique features of these microbes and their genomes are highlighted. To bring groundbreaking transformation in environmental, agricultural, and health research while benefiting the common people. To bring these results of the scientific results that we do in these journals, to bring this to common man, to the common Indian, that what are we doing and how are these microbial diversity in India benefiting the common man. And thirdly, coordinated by NIBMG, this program aims to make genomics data more accessible. So this is the, this idea of the One Health is now there, and where we know that not only humans, they are also, the human health is also connected to the animal health, to the environmental health. And in human, we have this human microbiome, which is more than 50% cells of our own, human body is of the bacteria, of the microbes, not only bacteria, fungus, and other microorganisms. So all the institutes, all the DBT and this Biotechnology Research and Innovation Council institutes are sharing genomes with NIBMG. All the genomes that they have isolated, if they have not sequenced, we are sequencing them. If they have sequenced, they are sending us the sequences, we are analyzing them, and then we are bringing out some interesting facts, sharing with their principal investigators. And this is the way it is going on, that sample collection and preparation that the institutes are doing. NIBMG is also taking part in this, and other institutes are also, they already have many samples with them. Then the bacterial whole genome sequencing is done from the strains that are isolated. This is not the microbiome. This is from the culturable bacteria. So this, the bacteria are cultured and the strains are isolated, and the DNA extraction is done. Thirdly, the whole genome sequencing is done either in that institute, if it is already the sequencing is done. Otherwise, they are sending it to NIBMG, and we are doing the sequencing under this initiative. And then we are analyzing them. We are using one analysis pipeline that we developed indigenously here, and we are analyzing them and we are sharing the results. If the PI is okay on the results, then we are actually, sharing the result in our NIBMG website. We have a section called One Day One Genome, and the more common man type things are being shared in the social media. We have made infographics, and the DBT individuals are also helping us. So we share those infographics in the social media, LinkedIn, Twitter, and Facebook. And more scientific things are present. If anybody wants to see beyond that, they can click on that social media. There is a hyperlink, and then they can come to the NIBMG One Day One Genome page and see all these antimicrobial resistance and other kinds of genes present in that bacteria, or some industrially relevant beneficial genes also. Now the achievements, coincidentally, today is the 150th day of the One Day One Genome project, and this we have been able to publish 150 genomes from these many institutes, and among them NCCS is the highest till now. And then NIBMG has also started doing these bacterial cultures and sharing them from the clinical samples. And then there are other institutes who are regularly, so there are many institutes who don't focus on microbes. So you can, evidently you can see there are 5-6 institutes who are generally contributing more and more bacteria. Every 7 days we are having a thematic, thing that on these 5 days NCCS will do. So they have a theme and they give all their bacteria, we analyze them and we share with them, and then if they are okay, then we share in the social media and our website also. And like that, we have now reached the 150th day with all these institutes. And while doing this initiative, we had these analysis pipelines for the microbiome, but we didn't have any high throughput analysis pipeline for analyzing the isolate genomes. So what we did in our lab, that we developed an analysis pipeline, and we call it Bharat. And this the acronym Bharat stands for Bacterial Hybrid Genome Assembly and Rapid Annotation Toolset. So what we are doing, we are analyzing both the short read and the long read together. The hybrid genomes we are doing, but mostly people are giving us short reads. So mostly the short read analysis is ongoing, but there is a module where we can also integrate the long reads. And this is a rapid annotation toolset because we integrated all the available annotation algorithms. And because we have to do one day, one genome, we don't have so much of time. So this is a snapshot of the Bharat analysis pipeline, and we call it 1.0 because we have also developed a second version 2.0 where we can do many other things, but those are not required for the one day, one genome project. So we are using this bacterial Bharat analysis pipeline 1.0. And here you can see the input files can be anything. It can be SRA, it can be nanopore sequence, FastQ, FASTA, anything you give, our analysis pipeline can take it. And then we have integrated several such algorithms, like there is an algorithm on Bactopia. Some of us are familiar with BVBRC also, but there are pros and cons of these separate algorithms. So we have integrated all of them such that the output file from one of them is the input file for the other. You don't have to do manual interventions. So once you give it here, and the NCBI, also when you submit to NCBI, there is this PGAP algorithm by which they analyze. We have integrated that also. So everything integrated, and you just input here and you get the output from here in a rapid way. You can identify AMR genes, toxins, and virulence factors, and it has integrated major databases like Abrigate, NCBI, CARD, RaceFinder, and VFDF. Now, we have sequenced and published these 24 bacterial genomes from our lab, but we welcome other PIs of NIBMG to also give any clinical isolated bacteria if they have. We will be happy to incorporate that. And we have also identified several of the anaerobes, some anaerobic bacteria from diabetic foot ulcers we have identified. One of them is a very dreaded AMR bacteria. We have got Neisseria brasiliensis. Now, another aspect of this one day, one genome project is in NCBI, we are submitting always all our genomes. But why in NCBI? Why not in some Indian archive? So now there is this Indian Biological Data Center. Our main aim is, if we are going to publish your genome in the one day, one genome, you have to submit it here in the Indian Nucleotide Data Archive. Now, if you have already submitted in ENA and NCBI, those accession numbers, if you can provide them, they will be connecting that. You don't have to submit again. So IBDC individuals are also relentlessly working with us day and night, and all these things are getting submitted. Only when it is submitted, then only we are publishing it in one day, one genome. Yeah, so these are some of the bacteria that we have also submitted. And we have checked the Bharat analysis pipeline results for the contigs and GC content, the genome size, coding sequence, and other things, rRNA, transfer RNA. And we see that when you compare with the NCBI PGAP, mostly they are similar. And also in some cases, they are giving better results because, these are updated ones. NCBI, when you see something has been analyzed, it's actually 3 years back or a couple of years back. So you are getting more contigs and, more significant results. Now, we have got some of these infographics are of very exotic bacteria by NCCS and other, things. And they are in high reach in both the Twitter, in LinkedIn, and in Facebook. And we have also now trained ourselves in making very interesting infographics, like the graphical abstract kind of things. In the BRIC one day, one genome page, you can see, you can go and click here, you will find all the bacteria for the 150 days. You can click them, you will find all the information present there. And you also have a YouTube video because on the BRIC Foundation Day, this was being highlighted, and the curtain raiser happened by the Secretary DBT. So this video is there. What is this one day, one genome, why it is important, and all the curtain raiser things. And lastly, our students have made a short video of few seconds that show. Thank you all, and I really thank my students, Shovik, Shankho, and also Dr. Shobhashachi Bhattacharya, and everybody like Bikram, Atanu, DBT persons, Dr. Vamsi, Gautam, our Professor Sagar Sengupta, our director is the nodal officer, and the scientific supervisors are Professor Arindam and me, and Nidhan and Dr. Alpana, they have helped us while we were trying to see that every day we have to upload it in our NIBMG website, how this can be done on a real-time basis. So at that time, it was seemingly very hard, but Nidhan helped us to find out an easy way around, and now we are following that. And Bikram and Atanu at that time helped a lot, that every day we are sending one genome to Atanu and he's uploading that. Now it has more or less got standardized, but when we initially started, it was like, we thought that how to make an impossible thing possible. And with all of their help, we are now being able to not only do this one day, one genome, but enrich ourselves also by building some analysis pipeline, building up a repository of, now we have more than 50 such clinical isolates from different human samples. And now we have archived them. We can also make a repository like they have in this NIRBI. Thank you. Thank you all.
Thank you, sir, for giving this overview on the ODOG initiative. Now I would like to invite Mr. Shankonath, ICMR SRF at NIBMG, to speak about his work, which is titled Environmental Arsenic Contamination and its Impact on the Human Gut Microbiome in India.
Hello. Okay, so I'll not take much time. So today I'm going to talk about the work that we have done in collaboration with another institute. So the work is on environmental arsenic contamination and its impact on the human gut microbiome. So human gut, you all know that human gut harbors trillions of microorganisms, and it has 100-fold more genes or genomes than us. So it is essentially called as the second genome. And gut microbiome plays a crucial role in maintaining the human host immunity. And dysbiosis means the imbalance in the microbiome. So dysbiosis in the gut is actually linked to several metabolic diseases like diabetes, chronic ulcerative colitis, and all those things. So environmental factors like exposure to heavy metals and xenobiotic compounds, they actually may cause disruption in the gut microbiome composition, and which can thereby result in the metabolic complications. And among the heavy metals, arsenic is the most crucial thing that has been found to exert detrimental impact on the gut microbiome composition. And there have been several studies in vitro and also in ex vivo mice models that have shown that arsenic exposure can actually alter the gut microbiome composition. So from the image, as you can see, arsenic can actually incite the dysbiosis in the gut, and it results in the oxidative stress, inflammation, organ damage, DNA damage, and cognitive decline. So all of those, as I have told, all of those studies have been done on in vitro or in ex vivo mice models. But there are exactly 5 studies globally that have done on focused on the microbiome, on arsenic, on the impact of arsenic in gut. So these are the studies. There have been studies from USA, Bangladesh, Nepal, and 2 studies are from China. And all of these studies have a sample size of 100 to 200, and they have all done 6ns rRNA gene sequencing for metagenomics approach. But all of these studies have not included healthy controls. They have just investigated the changes in the gut microbiome in arsenic exposed individuals, not compared to any healthy individuals. So in why we are studying arsenic gut microbiome, because in India, the 2 most important area, the most affected area are actually Bihar and West Bengal. And as you can see from this image, this white surrounded area is the Gangetic basin, and in this Gangetic basin, the arsenic contamination has been widely reported. And in West Bengal, among the 9 affected districts, Nadia is the worst affected district, with having arsenic concentration far beyond the permissible level, which is 50 to 60 microgram per liter. So that's why we have taken up this study, and our objectives for this study is to characterize the alterations in gut microbiome composition among adults chronically exposed to the environmental arsenic, and to further investigate the potential associations between these microbial perturbations and metabolic disorder related outcomes, and also to further investigate the alterations in microbial pathway enrichment profiles resulting from long-term arsenic exposure. So study design is, we have collected healthy control individuals from a same region, but Paschim Medinipur, which is not arsenic affected or contaminated region. So we have collected 50 healthy controls from Paschim Medinipur, from Garbeta 1 block, and from and the exposed individuals we have collected from Nadia district. So we have collected altogether 100 individuals. Among these 100 individuals, 50 actually have skin lesions in them due to arsenic exposure, and 50, although they are having high arsenic exposure, but they didn't show any skin lesions in them. So we have collected stool, urine, and also groundwater samples we have collected, and from these 3 samples we have done the estimation of arsenic using spectrometry, mass spectrometry. And from the stool samples, we have isolated the microbiome DNA, and from the microbiome DNA, we have done 6ns rRNA gene sequencing. So this is our analysis pipeline. We have done the V3, V4 sequencing, and from the raw reads obtained, we have actually gone to the Kain analysis pipeline. We have initially merged the reads, pair-end reads, because these are pair-end reads. We have merged the reads, and then we have gone for QA/QC based on these parameters. And after QA/QC, we have removed the chimeric reads. Chimeric reads often come from PCR duplication. And then after removal of chimeric reads, we have bin those reads into operational taxonomic units. So operational taxonomic units are devised based on or estimated based on the 97% sequence similarity. So these are essentially the operational taxonomic units. Like this, many operational taxonomic units we have estimated, and from each of these taxonomic units, representative sequences were taken, and they were assigned to Silva 138 database, which is the most updated Silva database for taxonomic assignment. And further on, the PICRAS2 algorithm or pipeline has been used, that is for the prediction of the microbial pathways. And for the downstream analysis, we have defined core microbiome. Core microbiome is the major microbial taxa that are present in our data. So that is based on those taxa that are having at least 1% abundance in all the samples. And further on, we have, so 6ns essentially gives us general level data, not up to species level data. But for the species level data, what we have done, we have taken the representative sequences from here, and we have used BLAST, and we have checked, we have assigned to those reads for the species, and only the reads which has shown 99% identity percentage for any taxa, we have taken them for species. So this is the gender and age distribution across all the groups, and we have, as I have already told, we have estimated the arsenic from the groundwater. GW is for groundwater, and also from urine and also from stool. So from urine and stool, we have seen that the healthy controls are having significantly lesser number of concentration in the urine and stool. And for groundwater also, as you can see, Paschim Medinipur has very less concentration of groundwater arsenic compared to the Nadia district, which is way above the limit, like 190 and 116. So the limit is 50. These are the total number of these are the total number of read actually we have obtained. That is 121 million paired end reads from all those 150 samples. And these are the parameters that we have obtained from the raw read. And as you can see, obviously, we have found more number of genera in the control individuals compared to the arsenic exposed individuals. And also the number of core genera is slightly higher in the control individuals compared to the arsenic exposed individuals. So then we have estimated the alpha and beta diversity. Alpha beta diversity means the intra-individual diversity of the microbiome composition. So within individual diversity, we have estimated. So these are the alpha diversity, and several indices we have also checked. This is Shannon, this is Simpson, this is Shannon, and this is Chow. These are all the estimates of alpha diversity. So statistics of alpha diversity. So we have seen that the healthy controls are having significantly higher intra-individual diversity. It means the healthies are actually having wide variation of the gut microbiome composition. But for the arsenic exposed individuals, the diversity has gone down significantly. And also we have done the beta diversity indices estimation. So this is based on Brake-Artis dissimilarity. And we have seen that the healthy controls are this separate cluster. These healthy control samples are actually separately clustering from all these arsenic exposed individuals. So it means that the healthy control microbiome composition is completely separated or separated from the arsenic exposed individuals, whether they are presenting any lesion or not. And then we have gone to the genera level data. We have seen that certain genera are having higher abundance in the arsenic exposed individuals, whether they're showing lesion or not. So Bifidobacterium, Streptococcus, Blottia, these are actually shown here to have higher abundance in them. And contrary, the good gut bacteria like Prevotella and also Faecalibacterium, they are actually decreasing in the in the arsenic exposed individuals. So I've given a table here. This is a lesion and this is without lesion. All these are arsenic exposed individuals. But all these bacteria, as you can see, they are having a similar microbial composition and their p-value is not significant. So it means that the lesional and non-lesional arsenic exposed individuals have a similar microbiome composition in them. So whether they are actually whether they're showing any lesion or not, so it is showing a systemic change in the gut microbiome. So as evident from the principal component analysis, the lesions and non-lesion samples are also showing similar microbiome composition. They are overlapping in the principal component axis. So we have gone to the species level also. So under those genera, Faecalibacterium, Prevotella copri, these are the two species of Prevotella and also Megasphaera. These species are mostly actually healthy gut associated species. They are found to be significantly depleted in the arsenic exposed group. And on the other hand, an obligatory Blottia that we have found, and also Streptococcus salivarius. So these species are significantly higher in the arsenic exposed individuals. So Blottia, Streptococcus salivarius, they are found in other locations of human body, but for gut, they are not that much of a commensal organism. Okay, so they are opportunistic pathogens for gut. So and further, we did have the arsenic concentrations from the gut, stool, and from the stool, urine, and also the groundwater. We correlated their concentrations with the microbiome abundances. From the urine, we didn't find any significant correlation, but from the groundwater and also from the stool, we did find a significant association or correlation with Bifidobacterium longum. So Bifidobacterium longum is a good bacteria, but what it indicates? It indicates a protective role, possibly a protective role that is played by Bifidobacterium longum because it produces short-chain fatty acids and it is a well-known bacteria that thrives in oxidative stress. Arsenic induces oxidative stress in gut. So further on, we have done the correlation network to see that how the bacteria, each of the bacteria in the arsenic exposed or in the healthy control, they are interacting with each other. So in the healthy controls, we are saying some specific cluster, like this is a Clostridium cluster, and this is a Bifidobacterium lactococcus cluster, and this is a Prevotella copri cluster. So these are gut-associated, healthy gut-associated bacteria that are showing a clusters. But in arsenic exposed, not that much of clusters or correlation or network between bacteria we have observed. So a complete dysbiosis has happened in the arsenic exposed individuals. And further on, the pathway analysis showed that two of the important pathways like formaldehyde assimilation and super pathway of so SAM biosynthesis pathway, they are important. These pathways are very important. They are found to be significantly enriched in the arsenic exposed individuals. So formaldehyde essentially is actually generated due to any heavy metal metabolism. In case of arsenic metabolism, it is also getting produced. So this formaldehyde assimilation happens in the gut bacteria, gut microbiome of the arsenic exposed individuals. SAM is also involved in DNA, RNA, and protein methylation on repair. So it is an indication of oxidative stress happening in the gut microbiome of the arsenic exposed individuals. So lastly, we also collected several metabolic factors, complication factors from the individuals. We did have the dietary information, the fat content of the diet, and also the metabolic complication data like obesity, allergy, ulcerative colitis, diabetes, and we have this data on a binary data, like if they are having or not. So we actually, what we have done, we have taken this data as a binary data, and we have done a binary logistic regression with the microbiome abundance, species level abundance, and we have seen the Streptococcus salivarius that we have already obtained significantly high in the arsenic exposed individuals. This bacteria is actually showing a significantly positive association, as this is a regression analysis. So significant positive association with the obesity as well as in ulcerative colitis. Allergy, we didn't found any significant association with any bacteria. And also that Blottia loti also is found to be associated with ulcerative colitis. So lastly, our conclusion is that chronic arsenic exposure actually led to the complete gut microbial dysbiosis with a depletion of good bacteria like Faecalibacterium, Premotella copri, and also but enrich the stress responsive taxa such as Streptococcus or Blottia. Microbial pathways we have also seen that are associated with DNA repair functions. And similarly, we have also obtained that lesional or without lesional arsenic exposed individuals are having similar microbial profiles. This implies a microbial dysbiosis in them, whether they are showing any lesion or not, and it is showing a systemic manifestation. And this could also, this shows a very good indication that the gut microbiome can act as a biomarker for the arsenic contamination area or arsenic endemic regions. So this work has been done with a joint collaborative research between BRIC and IBMG and CSIR and NERI. And this work we have published in Journal of Hazardous Material recently. Thank you. Happy World Microbiome Day.
Okay. Thank you, Shongoda, for highlighting the link between the environment and our internal ecosystem. So this brings us to the end of the formal sessions, but the conversations don't have to stop here. We have arranged some refreshments for everyone. It's outside. And happy World Microbiome Day to all.